Nag-ano
because
pa,
of the loss in
nagpapaturo po si
production.
Avi.
Because when the seaweeds are infected with disease, so they tend to
Parang may naiwan ng wire
sa
may signal na charger kasi it's like
fall into the second direction. So walang na-harvest ng farmers. And
okay ka lang?
you've isolated a bacterium which by 16S are already sequencing. We found out a microbe for SB and we named it the strain Alacoan because at that particular time, this is in honor of former U.S. Secretary Len Alabastro, and so we named the strain after her with these initials. But one interesting thing about this isolate is that instead of the usual colonies that grow on top of the agar, this one penetrates the agar and creates the substrate. And the culture medium is made up of carrageenan, a mix of carrageenan. So this suggests that this organism or microbe is capable of degrading carrageenan agar. So sometimes when we are actually thinking about the production of enzymes, how these organisms actually show this particular phenotype, and then always tend to actually think about the biology of the particular organisms. And these carried on actually. And I went to Sydney and worked on a seed disease model data system, which is this particular seed, Delicia vulgra, and an isolate coming from the disease Thallus, which is Nutella italica R11. So Nutella italica is formerly known as Bulgaria. And you can see here the Thallus being colonized by the R11 bacterium and form a biofilm on the Thallus. So, and in this particular photo, this is just show that show a facility at the Sydney Institute of Marine Science, which I also had a chance to work into. And this is where you can actually sterilize your seawater. So this is where we can do inoculation experiments, grow the seaweed in these tanks, and then seawater comes in that is sterilized, so you can inoculate it with electronic. It's a simple facility to think of, but it serves a purpose for us. And I am actually part of, so at UNSW, I am actually part of a group which we call Virulence, wherein we are looking at this particular seaweed disease model system and elucidate mechanisms. So I'm part of the small microbiology group, and I work on bacterial signaling systems and the use of attachment. Yung supervisor ko na is about that. And a number of people are actually working on seaweed ecology, so disease ecology. A couple of students are working on biology genomics, which is into the seaweed microbial community, and a number of students also are working on genomics. So let me just introduce you to the biofilm mode of life, which is a common lifestyle of bacteria in natural environments. So the bacteria would actually transition from a motile to sessile lifestyle under the biofilm. So typically in the natural environment, bacteria would be free swimming, and they would swim until they find a surface that they want to attach to, because they sense that the nutrients in there or any other biomolecules that would allow them to grow as well. Then once they find a suitable substrate, they commit and stick to that particular surface. And then once they, they are happy with on the surface, and then they begin to produce their exopolysaccharides and grow in numbers. But there will be a point wherein nutrients are depleted also, and the population or the cells inside the biofilm would detach and then swim out of biofilm. So the cycle continues. And for most of bacteria, this is the common lifestyle. So the stages of biofilm formation can be actually seen under the microscope. And the first stage would be reversible attachment, because again, when the cells find that a particular surface is not ideal for growth, then literally leave that particular surface and find another surface. And then irreversible attachment, stage 2, is where bacteria would commit and stick. Stage 3 would be they start to form micromolecular formation because they're conforming or excreting their extracellular polysaccharides. And then the biofilm matures. And lastly, stage 5 would be biofilm dispersal. Now, these are, as you will see later, that microscopy of biofilms can be seen done in the laboratory if we have flow cells. So these are just slides with channels, in this case, 3 channels. So you would already have 3 replicate biofilms ongoing. And then you essentially inoculate the flow cells and allow the cells to attach, and then add your medium, which is a continuous flow, and after a particular time, view the biofilm under a fluorescence microscope, in particular in the laser scanning microscope. And this is a nice, simple setup, but really nice setup. And a little bit of a nerdy setup, to say so, because if we tell you about tubes in the laboratory, then you can set up an experiment that looks like this, where you have here your culture medium, which goes into your flow cells by pump by a peristaltic pump, so at a particular rate, and then the used medium is received by a waste bottles or flasks. So in the laboratory at CMT, marami gumagawa sa atin. So we're looking at biofilms. And of course, we use a confocal laser scanning microscope to be able to see the three-dimensional structure of the biofilm. Again, sorry for the slide here, but the nice thing about confocal laser scanning microscope is that you can make slices of your images, then combine these images to form a three-dimensional image of the biofilm, and you can actually rotate the image using a particular software, and then you can see image left, right, and up. But in laboratories where definitely the confocal microscope is very expensive, but in laboratories that don't have this specialized equipment, you can actually grow biofilms on tubes and see the formation of biofilms on tubes at a particular time. So essentially what you do is test tubes, and then you see you let your organism grow, and after some time you see pellicle forming, and after some time also you see that a nice biofilm that's actually formed on the tube. And what you can do with that is you can actually readily remove the spent culture medium and then stain it with crystal violet. And then once you stain it with crystal violet, you can use crystal violet by ethanol, and then measure it under its smoothly spectrum. And you can have absorbance values that would tell you the amount of biofilm, or correspond to the amount of biofilm that's in tubes. And of course, if you're dealing with number of assays. So you can use a 96 well plate and or a 12 well plate or a 6 well plate, again depending on your number of replicates, for example. Similar, you can go your biofilm here and determine biofilm fermentation, for example. Now, at that particular time when I was in SIG, uh, this particular molecule is actually in attention. So cyclic diGMP, this particular molecule is a nucleotide. So you have here your base, you have your phosphate and ribose, and these are just synonymous terms referring to cyclic diGMP. Cyclic di-guanosine monophosphate, cyclic di-guanosine 3,5, cyclic di-guanosine. And this molecule is gaining that particular attention because it is a molecule that serves as an intercellular signal molecule. And pools of cyclic diGMP can actually be made inside the bacterial cells, and these molecules regulate certain phenotypes of the cells. So the synthesis and degradation of cyclic diGMP involves proteins or enzymes, which we term as diguanidine cyclase. Two molecules of GP are cyclized by a diguanidine cyclase, which contains a GGPF protein. And the degradation of cyclic diGMP involves phosphodiesterase, and cyclic diGMP can be degraded into this molecule GGG, and another phosphodiesterase can actually be degraded into 2GN. There is another phosphodiesterase with a different domain, so HTGYP domain, that hydrolyzes cyclic diGMP. But for this particular talk, we'll only focus on those proteins that contain GGPF protein. This became interesting because a number of studies have surfaced that cyclic diGMP controls transition from a motile to a sessile lifestyle. In free-swimming bacterial cells, cyclic diGMP is low, and then once they attach to a surface, then the pools of cyclic diGMP inside the cell increases, same as with when the biofilm actually is and matures. But upon cell death and dispersal, cyclic diGMP is we see here a diagram showing Pseudomonas aeruginosa is model system used to study cyclic diGMP. Significant analyses have been done in these organisms, including Vibrio cholerae. But let me just point to how cyclic diGMP controls certain phenotypes in Pseudomonas aeruginosa. So you see here a cyclase or a receptor protein which receives particular signal, and this WSPR is actually a cyclase which produces cyclic diGMP. The cyclic diGMP binds to a protein complex and FEB, and this regulates two of the phenotypes in Pseudomonas aeruginosa. So if the level of cyclic diGMP is low, this protein inhibits the production of L polysaccharide, which is a polysaccharide component of Pseudomonas aeruginosa, but allows the expression of flagellar protein. So at low cyclic diGMP, motility is enhanced. Under high cyclic diGMP, so when pools or when the molecule binds free end protein, expression of flagellar genes are repressed and allows the production of extracellular polysaccharides. So you can see actually the modulation of the cyclic diGMP. And this happens again when cells are actually sensing their environment. In Vibrio cholerae, when they are in the aquatic environment, they exist as biofilms and exhibit high cyclic GMP. And when they are free swimming, again, they will exhibit low cyclic GMP. Then when they go inside the host, for example, they infect yung
Comfortable ka ba nagsasalita na may nakikita ang tao sa
human, na-infect na Vibrio cholerae. So attachment would require the intestinal structures would actually require high cyclic GMP, and then the production of virulence factors would require medium to low cyclic GMP, and high cyclic GMP also be required for the infection. And this diagram shows that a number of type 1 cyclases from cyclic GMP are involved in the regulation of the Vibrio polysaccharide. So once cyclic GMP is produced, it binds to the VPSD and allows the expression of the Vibrio polysaccharide and forms the above. Now, again, I've mentioned about this interest in cyclic GMP because the sequence genomes, so at that particular time, the genome sequencing is already at high base, so madami na nasi-sequence ng genomes, and even metagenomic data would tell us that there's a multi-species GGTF or EF domains. And you can see from the protein family database that a good number of bacterial species would actually have GGTF domains in their genomes as well as EF domains in their genomes. And it took some time to actually look at, because in 2009, when I started work on cyclic GMP, around there's only about 600 species of scattered species of bacteria which has EL domains, and definitely we expect that up to this year, a number of the species with genomes containing or proteins genes encoding for proteins with EL domains has actually similarly with GGTF, so about 681 species in 09, there was about a larger chunk of species in these domains in and what is interesting about these particular proteins is that proteins containing GGTF or EL domains contain sensor domains such as PAS, GAF domains, MACE domains, or CHASE domains, and these would have certain functions. So PAS, for example, would allow for oxygen sensing, redox sensing, or light sensing, the BLAF figure. So the BLAF sensor domain would actually sense good light. GAF domain function for small ligand binding. The MACE is a membrane-associated sensor, and the CHASE is an extracellular or periplasmic sensor. So because of this, sensor domains that indicate that these particular proteins are actually involved in bacterial secretions. So in many bacterial species, nag-differ yung number of these particular proteins. So may mga species like this Vibrio cholerae, which has about, I think this is about 60 plus proteins containing GGTF, and an EAL domain, a sole EAL domain, and of course the HBG. And of course these protein domains are actually associated with a number of sensor domains as well. Prediction of domain structure is easy. So given a particular genome, we can get the nucleotide sequence and the protein complement, and then we can just enter it in this particular tool, smart database, and then uh, yeah, the result upon query would give you that domain structure. This is where yung mga bioinformaticians natin and computational biologists would come in handy because yeah, if you individually retrieve individual genes, for example, your coloring, all retrieve about 60 genes and then input them into the smart database. But with the help of bioinformaticians or computational biologists, a single line of code or program can actually just allow you to do the predictions at with all of these. So the question, based on these studies, the question that we have was that is there a role for cyclic IgMP in red algal pathogen, red algal associated pathogen of LN11? And again, we've sequenced the gene, so a chromosome and a plasmid, and we found out that there are certain virulence traits that actually allow R11 colonize and attack the sequence, one of which would be type 4 fimbriae and cellulose fibrils, which is involved in lesion colonization, invasion of algal cells, wherein there are genes and for acetic acid synthesis, glycan synthesis, defense against algal oxidative burst, and CMIDs would have this defense system, so we call algal oxidative burst, and R11 has enzymes that actually counteract oxidative burst. So superoxide dismutase, catalase, peroxidase, and other peroxidase related. And of course, R11 also has genes that encode for hemolysis type proteins and RTX toxic tyrosine. So, based on its is cytokine GMP involved in the regulation of these virulence traits. So, yun, siguro the Oragon in me actually thought, okay, I know that R11 has this number of genes. So it took inspiration from this particular paper, the genetic reductionist approach for dissecting individual roles of GGDF proteins within the cyclic IgM signaling network in Salmonella by Solana and co-workers. But the idea behind this particular paper was actually to construct a Salmonella strain in which all genes, including GGDF domain proteins, are deleted, which is about 12, and then restore each gene one at a time. So it took a lot of convincing from my supervisors to actually do this type of work, but definitely I did the work, and of course there were some challenges and extra challenges that